Hello, I'm Chris McCarthy from the American Chemical Society. Welcome to this news briefing from ACS Spring 2021. We're joined today by Dr. Ivan Efremov from Fulcrum Therapeutics Incorporated. He's working toward a reliable oral treatment for sickle cell disease. Dr. Efremov?
Yes, hello. Thank you for giving me this opportunity to talk about the work done at Fulcrum on identification of FTX6058, which is a potent orally bioavailable fetal hemoglobin upregulator for treatment of sickle cell disease. We can go to the next slide, please. That's just the title that I described, and thank you for advancing the slides. Fulcrum Therapeutics is a biopharmaceutical company that systematically evaluates small molecules that are able to modulate gene expression. Our goal is to treat genetic diseases at their root cause. To achieve that, we evaluate perturbations such as chemical probes or functional genomic tools such as CRISPR libraries in cellular systems of higher levels, the models of disease. We also do that in clinical samples when available. We obtain very data-rich data sets, which are interrogated by our computational engine, which takes advantage of machine learning methods, and the outcome is identification of highly validated biological targets. And one could argue that choosing the right target is the biggest predictor of future clinical success of the corresponding program. In addition to targets, we also get insights into potential toxicity effects and translatable biomarkers. If you can advance to the next slide, please. Thank you. So here, we're talking about finding a treatment for sickle cell disease, which is a devastating disease that has, it's a systemic disease, which has effects, a significant effect not only on the quality of life, but also on the lifespan of patients. And disease is actually caused by mutation in adult human hemoglobin. The resulting mutated hemoglobin, which is called sickle hemoglobin, is polymerizes in deoxygenated conditions, which leads to distortion of morphology of red blood cells. They adopt this characteristic sickle cell shape, hence the name of the disease, which leads to the disease manifestation called, for example, vaso-occlusive crisis and hemolysis of cells. So, interestingly enough, adult hemoglobin is not the only functionally capable form of human hemoglobin. There is also fetal form, which is typically not produced after birth, only in development. But there is a proportion of sickle cell disease patients that have this condition, which is called hereditary persistence of human hemoglobin. That is, they produce the fetal form in adulthood. And interestingly enough, these patients have much decreased severity of symptoms. And the higher the proportion of fetal hemoglobin that they produce, the milder and the fewer symptoms they have. And the levels of HbF, that is fetal hemoglobin, approach 30%. These patients often present as asymptomatic. So this condition really provides a genetic validation for the disease-modifying approach for treatment of this disease. And so the Fulcrum's goal was just that, to phenocopy hereditary persistence of fetal hemoglobin by upregulating fetal hemoglobin form with a small molecule. Next slide, please. Thank you. So to achieve that, we did a screen in HUDEP2 embryonic progenitor cells. We screened chemical probe library and the CRISPR library. The hit validation was done in CD34 positive cells differentiated into erythrocytes. In the course of this work, we identified some targets known in the field, but also we identified 6 novel targets. And of them, PRC2 complex was particularly interesting because it was found both in the compound screen and in the CRISPR screen. So we have high confidence in this target. And we chose a subunit of this complex named EED as the focus of our chemical program. The promising lead series was identified using a conformational control concept, which I will be describing in more detail later in my talk later today. And this, so we were able to rigidify the bound conformation of the more flexible EED ligands. We used structure-based drug design and multi-parameter optimization to expediently identify our development candidate, FTX 6058. It's a compound with a very promising with a very promising profile. It significantly upregulates fetal hemoglobin in CD34 positive cells, but also in the Towns model of sickle cell disease. Regardless of the basal level of HbF, we observe 2 to 3-fold HbF induction, which we see as very promising. In addition to these promising levels of HbF induction, we also observe a pancellular distribution of fetal hemoglobin, which really phenocopies the situation in hereditary persistence of fetal hemoglobin, where both HbF is upregulated and the number of F cells is increased. We think that this pancellular distribution achievable with our development candidate bodes well for the future success of the program. In conclusion, we identified a novel target using our product engine. We developed promising and a compound with impressive pharmacological profile, which has potential to be disease modifying. One thing I neglected to mention that both the preclinical PK, but also emerging PK from the human clinical study supports once-a-day oral administration, which obviously offers significant convenience to patients. At this time, we are actively enrolling healthy volunteers in phase 1 clinical study. And thank you for your attention. I will be happy to answer any questions.
Great, thank you so much. So if we have questions from our viewers, please state your name and affiliation in the chat window when you ask your questions. And I'll start with a question. How common is sickle cell disease?
Unfortunately, it is quite widespread around the world. The prevalence varies depending on the territory. In the United States, there are, if I'm not mistaken, about 100,000 patients. Most of them are African Americans.
And then, so what is the difference between the fetal hemoglobin and the adult version?
So the difference is subtle, but the way it manifests in vivo is that fetal hemoglobin is produced when human being is really in the fetal form, is not born yet, and really tapers off after birth. And adult hemoglobin is in turn follows embryonic form hemoglobin, then fetal form of the human hemoglobin, and then it really takes off when after birth. So it is interesting that some people, due to yet another mutation, actually experience this condition of hereditary persistence of fetal hemoglobin, which is really benign in its presentation, but is capable of ameliorating sickle cell disease symptoms.
So the mutation that causes sickle cells only affects the adult version of hemoglobin. It doesn't impact the fetal version. Exactly,
it's actually limited to the globin that encodes the adult form of fetal hemoglobin.
Great. So could there be any harmful side effects? I think you said it was benign for adults to express.
This is a great question. I have to admit that I'm not a clinician, so I cannot answer it with 100% certainty. But from my experience with the field, I haven't heard of any detrimental effects.
Okay. How did you get the idea to tap into the hemoglobin maturation for sickle cell disease treatment?
Excellent question. The idea actually is well established in the field, and it is hardly surprising because the hereditary persistence of fetal hemoglobin does provide genetic validation for this disease-modifying approach. So the clinical idea to use this approach is not novel, but finding this molecule has not been straightforward.
And how did you isolate the molecule?
So the molecule, again, our approach is first to identify a target that could modulate a disease. And we've done that in the course of using our product engine. Once we had a target, now we can embark on to the identification of chemical matter, basically compounds that could modulate the target. And we explored a number of approaches, virtual screen, de novo design, structure-based drug design. But eventually the series was identified using a conformational lock, and as I said, when we used chemical starting points which were flexible ligands, and we used a conformational constraint to lock the bound conformation.
Great. We have a question from Megan Brooks from Reuters Health, who asks, how is sickle cell disease treated now, and how could this differ?
Excellent question. Again, I'll do my best answering. I'm not a clinician, but standard of care for many years was treatment with hydroxyurea, where the efficacy was, I would call it suboptimal, and definitely not patients responded to that. This area gratifyingly receives a lot of attention now, so more treatments come on board, which is obviously great for patients. The benefit of the upregulation, of the approach that takes advantage of upregulation of fetal hemoglobin, is that addresses both the hemolysis and the anemia.
So it's more comprehensive than just treating symptoms. It goes to more.
So it's a disease-modifying approach which can prevent both, anemia and a complication resulting from organ failure.
Okay, great. So we have a question from Annette Bernidi from BioWorld. And she asks, can you talk about the advantages and disadvantages of oral meds versus more permanent solutions like bone marrow transplant or gene editing?
Right. Obviously, gene editing is a new frontier in drug discovery, which is great. I firmly believe that patients should have many options. Small molecule approaches do have their own advantages. So obviously, there is no special conditioning is required. The dose can be easily adjusted, and if unfortunately a patient does exhibit some side effects, the treatment can be easily stopped. In addition, so oral administration obviously has a benefit of convenience and has obvious advantages for patients in the US, but also makes access to this potential medication enabled in other parts of the world.
Thank you. So how long does it take for the patient when they start taking the drug to upregulate the fetal hemoglobin production?
So I cannot answer this question. Hopefully we will learn it from the clinic. However, in our in vivo models, we observe effect after 2 weeks of treatment.
2 weeks, okay, great. Do you anticipate any side effects from the FTX 6058?
That's the key question. And of course, any drug should be both safe and effective. So we were very satisfied with our preclinical toxicology studies, and FDA certainly agreed with our conclusions, thus allowing us progression in the clinic. And I would mention that we are moving into healthy volunteers in a non-oncology indication. So our preliminary toxicology work supports progression into clinic.
Thank you. Could this new drug be used to treat conditions other than sickle cell disease?
That's a very tantalizing suggestion, and there are certainly indications that other diseases may benefit from this treatment, such as beta thalassemia. And
beta thalassemia, how is that? What is that? I mean, people may be more familiar with sickle cell. What is not unable to say that right now, but how does that? What is that? How does that disease express itself?
So I'm a little bit on shaky ground because I am not, again, I'm not a clinician. I've been focusing on sickle cell disease. It's caused by a different mutation. It's also can be addressed by, we think, by adjusting gene regulation. So from that perspective, it is very similar to sickle cell disease. And there is certainly literature out there and supporting information that fetal hemoglobin can be can be suitable for treating some beta thalassemia patients. I have to indicate that beta thalassemia is really a very broad term, and there are different diseases under this umbrella term.
Okay, thank you. So what are the next steps for this research going into the phase one? Obviously is a big part, but what do you see down the line?
Well, we are very excited about being in phase one. So we will get a sense for the, obviously for safety. That's the whole point of doing phase one clinical trials. Hopefully, we will see target engagement in our studies. That's one of our goals. But this question is very relevant. Our next big goal is to transition to testing, to evaluating this compound in phase one in sickle cell disease patients, where hopefully we will be able to see upregulation of fetal hemoglobin. I have to say that seeing upregulation of fetal hemoglobin in healthy volunteers, while possible, but it's not necessarily likely in a short-term study.
Thank you. So what take-home message about this research would you want to leave with our viewers?
So we're excited that we developed an approach that can follow this attractive clinical hypothesis that upregulation of fetal hemoglobin can treat sickle cell disease. Because again, that condition provides hereditary persistence of fetal hemoglobin, provides genetic validation for this approach. We are excited that this approach can be disease modifying, addressing many, if not all, symptoms of the disease if the level of upregulation is sufficiently high. And the convenience of oral dosing is quite important around the world, but also in the US. And the fact that we advanced a clinical candidate that can be administered, we hope, once a day in a low dose, I think speaks to the quality of the molecule.
Thank you so much. The archived version of this session will soon be posted at www.acs.org/ACS Spring 2021 Conferences. And please join us for our next press conference Monday at 2 p.m. Eastern. Thank you.
Thank you for your time.